I'll start off and ask a question to Terry. And my question is, has there been research that has been done on some of the oldest samples? Because I think as most of us know that when you freeze things, it just slows down the degradation process. It doesn't completely stop it. So what kind of information would be lost? And is there research that helps preserve those tissue samples?
So you mean, has there been research on the tissue samples themselves to make sure they're of high quality?
Of their high quality, yes. Yeah, absolutely.
There's been lots of that with our Arctic investigation, specimen bank being part of the CDC itself. There's been lots of looking at that, and apparently now the CDC in Atlanta is looking at our standard because of our lab and our field station in Anchorage to be copied. As for research from the samples, there's been quite a bit of that, some of it with permission, some of it with a waiver of permission. One of the most recent studies I can remember is looking at old collected specimens from women of childbearing age about 40 years ago, so women from 19 to 34, and looking at the vitamin D levels of those women and comparing it to the vitamin D levels of our children in our primary care clinics. And that's been useful research on the specimen bank done by Ross Singleton and team.
Excellent. And I would like to direct a question to Dr. Henderson. Vince brought up a really great topic about diversity of the workforce. And you're doing some really great, wonderful molecular research right now. What do you think are some of the most effective ways to recruit American Indian scientists to become more engaged in this field of research so they can have an impact on their communities?
You know, in this particular field of research, it's, you know, just as we understand the journey that our Creator has for us, you know, not all of our relatives are fated to, you know, to work in this realm. It's usually the case that a budding genomic scientist isn't exposed to population-based approaches or is asked to consider population health, and usually will not have a clinical background, so will not be able to have a career that's informed by clinical experience either. I think one of the things that has to do is we will have our relatives, our youth will naturally flow into molecular biology and genetics, and hopefully we'll be able to expose them just, you know, through a more population-based lens. That's what it's going to take, I think, to really explore and develop the kind of studies that might have more timely benefit to us than not. One of the things we've been doing now, so aside from that, the University of Colorado, Denver, and the University of Washington have been partners since 2000 on a research center for minority aging research grant initiative funded by the National Institute on Aging. We've taken the funding we received from this and developed a stay-in-place 2-year crash course in how to conduct research, the nuts and bolts of how to conduct research. Very little theory. You know, these scholars have their theory from their principal training. This is about how to conduct research. It's a 2-year program. We meet 5 to 6 times a year. Secondary data analysis takes up year 1 along with a manuscript developed out of that, which helps to inform a primary care data pilot collection process in year 2, which then helps to inform your exit deliverable as you exit the 2-year experience, which is your next grant proposal. Whether it's a R01 or R21 or a K award has been real popular, we've learned. We're now in our 8th cohort. We've been doing these for 16 years now. We have 42 postdoctoral American Indians who have come through this program and successfully completed the program now, who collectively have I think, published 380 or 90 manuscripts, have $48 million in grant funding, have significantly helping to move the bar, if even in a tiny increment, in terms of our representation in the NIH-funded, investigators pool, as it were. So those are a few random thoughts.
Wonderful. Do we have any comments from? I can't see over the bright lights. Or can you stand up and go back to the microphone in the back so the people who are seeing this on video cast can also hear you?
My name is Phyllis Massey. I'm enrolled in the Oakland, Missouri tribe and a member of the Cherokee Nation. I work at the Huntsman Cancer Institute in Salt Lake City, Utah. And I have several comments and some questions. The first one that comes to mind is we have talked, as we talk about research and we talk about genetics, and as we've come through this program is the Havasupai tribe. It comes up over and over and over again, and it's something that if you're involved in research and you go to the tribes and you talk with PIs, or you coordinate research with PIs, you have to deal with. The one thing that I don't have an answer to, and I hope you do, is why do we not have answers to what happens to unethical principal investigators who work with our people? Nothing happens to them, and they continue to get funded. NIH, NCI, every single federal institution, if you look for those researchers, they continue to get funded. Why? That's my first question.
Anybody on the panel want to try to tackle that one? I think that is something that is beyond the scope of our panelists, and it's a really important question. But I think that what we can do here is take away the positives. What are some of the things that we learned from that? And
But that is a very important question. Because if we, as Indian people, want to do research, and we want to earn the trust, which I have tried to do, and I want to bring other researchers to the table, and I want to explain that, yes, this happens. It's not about the research. It's about the researcher and the ethics. We can have all the best researchers that we want. But if I am going to help you come to my tribe to work, if you do it wrong, you are going to ruin it for every other researcher that comes after you.
That's a positive. That's a good question, and I'll jump right in there and try to find a channel in the middle of the river. This question about researchers, and I think you called got to get my glasses on as I've aged. Why do we continue to fund unethical researchers across the funding course? I think that Indian country is a really, really small place. And although it covers all of North America, if you get to know people like Canadian friends and Midwestern friends and Southwestern friends and farmer friends, if a researcher suddenly pops into your part of the world, or your part of Indian country, it's very, very easy for us to pick up the phone or an email, or even now we tweet to each other, for gosh sakes, and ask if they say, I've done research with pregnant mothers and breast milk in the far northeast. I know exactly who to pick up that phone and call. And you will too, as you continue to do good research. And so what happens over time is the opportunities to improve the researchers that have hit the sandbar have less and less to do with tribal nations in America. That's kind of sad. I watched them go to the Congo and to Latin America. That's kind of sad. I don't know many people down there where I can spread the word. But I think if you continue to set a good example and look for our resiliencies, things we've done, I think that's changing.
I agree. I hope so.
I just wanted to also add a little bit to your question. For the human subjects protection realm, there are steps that are available if a researcher is acting in an unethical manner. It, however, is based on the institution. So the institution is responsible for actually holding the researchers accountable. And if they are unethical, they need to report them to the Office of Human Research Protections. And depending on what the egregious acts are, OHRP then actually, they're known through the research world that they are acting in unethical manners. And this can actually then impact their publications. Publications can actually be withdrawn. Their funding can be taken away, and this is communicated to funding agencies. So there are mechanisms in place to punish unethical researchers. There are ways to blacklist them. Again, it's dependent on the institution and how they go about pursuing those different steps.
That's true, but however, in this particular case, all she had to do was leave the institution and go to another one and start all over again. And that's generally what they do.
I'm going to tag right on to Francine. You know, there are OHRP rules, there are funding rules, and you know about those. Another place that you can find friends is in the compliance world. I never thought I'd actually say this. But as you attend the meetings, get to know those people that run the University Office of Research Integrity at places like Pennsylvania. Get to know them personally, be very accessible, like Francine says, and really and truly, it takes a single phone call to the University of Pennsylvania compliance official to stop a study until it's put right back in the middle of the river.
No, I mean, I have done that. I have been the person that has gone to my office and stopped research for my own PI. But that's not what I'm on the fence too. I have helped with HNPCC, with tribal members, with the Eastern Shoshone because it helped. But then like I said, with an ethical researcher, once they have been put on the table, you know, their wrists slapped, all they do is go on to someplace else. And we don't do anything about it. And as we move into genetics, which is very important, especially when we come to therapeutics and pharmacogenomics, it's so important that we participate. But once they get our blood samples, and if they want to do something else with it, and if they're unethical, that's when I start going, wait a second, all over again. But anyway, I just want to put that on the table because on a federal level, we don't do anything about it. We just, they continue to fund them. I've done my research on it. I've looked, we can do it at an institutional level, we can do it at a state level, but at a federal level, they continue to fund them. So, Dr. Francine, when you were going through the review for the informed consent, one thing that I would like to add is I started, particularly with the informed consent process, I took probably 6 months to educate the tribal community that we were going into on the informed consent that we were using. Doing that on a weekly basis, evenings and weekends, so that they understood very clearly the research that they were getting into. And we gave monthly updates, community updates, so that the tribe knew what was going on, the progress. Because as it moved into genetics, as we move into genetics, there's a lot of information that they really don't understand. They want to participate. There's a lot of language, there's a lot of new words that if they don't have the meaning of, or if they don't grab the concept of, then they can't ask the question about. And so it was no longer just a quarterly, once, 4 times a year. It was every month. Even if there was nothing to report. And also, we put a Native person at every step. My assistant and I, we became the phlebotomists because we just didn't have any Native phlebotomists to draw biospecimens, so we became the phlebotomists. So we could have a Native person doing the informed consent, drawing the blood, doing the questionnaires, doing it. And it's really great because, as you were asking, how do you make a Native researcher? How do you get them involved? You go to the university and you say, come spend a weekend with us. Get involved in the study. And now we've got some kids who are doing some things that they never thought they were going to be doing. And we've got them in the labs. So it was also by building trust, by having someone Native all the way through, and being a graduate, student working directly with the PI, because we were involved with biospecimens and it was genetics.
So you bring up a really good point, and I think this is something that our partners can take note of, is that having peers and role models of Natives students is extremely important. So we saw the diversity numbers from the NIH and the investigators that it showed that there were absolutely no Native American NIH tenure track investigators right now. Well, that's a big problem. How are we going to build upon our Native students? How are we going to give them the confidence to pursue such a daunting endeavor? And you're right. So I think that this is a really important issue that we should point out.
Every time you go out, it's go to the undergraduate students and just bring them out.
Mm-hmm.
And you can do it, and it's so easy to become a phlebotomist. I mean, it's so easy to participate. That's all I have to say.
Thank you. Yes, sir.
Good morning, my relatives. I love that phrase. That was a really nice way of putting a greeting in the context of biology that we're starting to understand the genomes. I'm Nathan Pearson from the New York Genome Center, and coming here just to meet folks and to really get to hear the perspectives today. I wanted to ask 2 quick questions. Dr. Gachupin, how do you say your name, by the way? Okay. So I love this insight that a pedigree can be much easier to identify within a tight-knit community, throughout the whole community, if folks know a bit more about the scant information that's provided, say, in a paper. So you mentioned ways to try to avoid that pitfall. And I wonder if you can help us understand some of the practical ways we could do that in working with people to publish data that preserve that balance the need for individual privacy and autonomy with that for being able to understand scientifically what's going on with the data. And then for Dr. Henderson, I had one quick question for you about many of the drug response data that you showed was really interesting, and the plants in question, so tobacco and cocaine, or coca, both are American plants originally, and so I'm curious about what we know yet, if anything, about the diversity of how people respond to those, depending on how far they live from the places where, say, tobacco or coca were domesticated, or how recently they were introduced, say, in the Great Plains. Thanks.
My mic's not working, so I have to borrow Terry's. So really a way to really help to keep your data confidential or anonymous, depending on what the agreements with the community are. is dependent upon the study, because for some studies it may be involving a family and you're studying something that's hereditary in transmission or something. And really you're trying to protect that family because that information may not be known throughout the community. And so really working with the family and the community in how best to protect the individuals involved in the project, but still maintaining the scientific integrity. You want to make sure that you're talking with the community about how they want to have place names, tribal names, identified within the manuscript itself. Some tribes want to be recognized as a tribe in the Southwest or an American Indian community. They may be okay without actually having the place names and the tribal name in publications. It really is making sure that all final copies of manuscripts are reviewed prior to publication and making sure that everyone's okay with however those descriptors are going to be included within the manuscript itself.
To use a concrete example, I showed the SYMP2A activity and the striking results for South Dakota, which came from 2 Lakota tribes, and one of the 2 tribes redacted their name out of the draft slide set that was given at the Society for Research on Nicotine and Tobacco's annual conference in Seattle this past February. And so as a PI, I made the decision to redact the second Lakota Sioux tribe's name as well. So, we're real happy. So that was a slide set. We'll see what the issue, what the conditions will be for a manuscript. Will definitely be produced off of those particular findings and a number of different manuscripts. And I don't really have any stake in the game. It's not a point for me to advocate that the tribes allow their name to be used. At least not for me. So I'm fine with whatever the tribes decide. But we do have a very good, very transparent discussion about that. To the second question, I'm very much less familiar with the history of Eastern Seaboard tribes. Broadleaf nicotinia species of tobacco, I believe were imported into this country. I don't believe that they're indigenous to the United States. That we do a tremendous amount of tobacco policy and control work out of our center, and I believe that that's the case. None of the tribes west of the Mississippi River that I'm much more familiar with used nicotinia species of tobacco at all in the ceremonial ritual uses. We may have called what we smoked in a pipe bowl tobacco, but it was not nicotinia species tobacco. In fact, it wasn't tobacco at all. I know for Lakota people, our traditional tobacco, or chanshasha, was the scraped inner bark of the red willow, which was cut with a little bit of bearberry, dried bearberry. That was mixed up, and that's what's used traditionally in ceremony and ritual. However, much of our research, much of my wife's research, Dr. Patricia Nez Henderson, is focused on the history of how commercial tobacco came to replace our traditional tobacco in ceremony and ritual, in addition to its uptake in a secular manner in day-to-day life. So, what do we know? So, what do we know about this? We know very little about the effects of smoking commercial tobacco on native peoples. Some of this SYMP2A work has been done in a few other tribes in the United States, but only a few, maybe 2, maybe only 1 other than our study here. So these are early days in examining the genetics of smoking and nicotine metabolism in our relatives.
Excellent. More questions from the back?
I'm Yate. I'm Nana Garrison from Navajo Nation and currently at Emerald. The question I have is inspired by 2 of the panelists, Terry Powell and Francine struck by the words that, the care that you take in talking about terms like discarded, destroyed, or biohazards in the context of biospecimens. And I was wondering if you can talk a little bit about how you frame the conversation about either returning these samples to communities, or how you talk about handling these biospecimens after a project is completed, or when a person requests those samples to be returned. Rather than using the words destroyed and discarded, how do you frame that conversation?
Sure. Can I go with the first? Okay. So depending on the community, again, this is all relative to individual relationships that a researcher is establishing with the community they're working with. But a lot of times it's really the tribe coming forward and saying, we want control of these specimens, and part of that control involves the return of those specimens. And the return of the specimens can be dependent upon somebody's death, for example. We can keep that sample for as long as the person is living, walking on this earth, but when that person dies, we want those samples back. Or it could be that the tribe is participating in a project, but they terminate the project. They don't no longer want to be part of the project. And that's a very real scenario, because tribes have changes in leadership, and there could be a tribal leader who's very pro-research or very pro-studying this or that. Another leader comes in and they stop it, and they say, no more. We don't want to be sharing our samples. We don't want this to be happening. And at that point, samples could then be returned. So it's not individual based at that point, it's the whole project, and anything that may have been acquired up to that point being returned. And that's one of the examples I gave earlier, is that's happened to one of the tribes I worked with, who were in mass, they wanted all the samples returned. And that's when we engaged a local institution laboratory to help us to dispose of those samples properly. And again, you can't just take that specimen and hand it over to somebody. They need to be disposed of properly. And it's really educating the community about what that means and what options they have available to them at that point. And so it's working kind of on an individual basis. And by working with that community, you learn what the respectful way is of communicating with them, and what is considered to be the proper terminology, the proper sequences of doing things. And they're trying to educate you about who needs to be involved in that process, and making sure that those correct authorities are present and are giving their input into that whole process.
Up in Alaska and across a lot of Indian country, we're an oral culture. And so we try to build our relationships with our research partners and communities. And we've learned the hard way in northwestern Alaska that what our tribal leaders there thought they understood from a large pedigree study across the western part of our state, and what was written down were two different things. And so I suggest to tribal leaders that they have a policy before embarking on a project as a community or as a tribe. Oftentimes ours are regional because our numbers are small in each of our rural communities. Ahead of time, that's agreed upon with the researcher, with the funding institute, and that has proven to be very, very successful. I was in Phoenix 2 or 3 weeks ago for Native Research Health Conference, and there's a large nonprofit genomic company there, and they're working with a tribe who likes to be named. And Dr. Trent stood up and gave an hour presentation about all the materials it took to get in place for his company to work successfully as a collaborator and a neighbor with this little ways away from Phoenix tribe. And the material that the homework they've done and the relationships they've built are really going to pay off in the long run for this nonprofit, because the tribe kind of decided what they wanted research on. It wasn't what this large institution thought. And they just switched gears and moved on with it. And I think it's ahead of time, and it happens continually. Be available. Answer the questions.
Thank you very much for the questions. We have about 10 more minutes left, I was informed, before we get the hook off the stage. So I'd like to try to get as many questions in as possible.
Hello, Terry. Okay. My name is Dorothy Aguilera Blackbear, and I'm Choctaw. Thank you so much for sharing your knowledge with us today and the work that you're doing. My question is kind of one that deals with traditions and holistic ways of taking care of our health and wellness that isn't identified necessarily through the kinds of research that you're doing. I'm wondering how is that intersecting in terms of the research that you're doing with some of the tribal people that you're working with?
I work with our relatives in a really highly participatory manner. That this is a particular kind of a study. This is a follow-up study of a large cancer study that we conducted several years ago with over 5,700 of our relatives in communities all across the country. And one of the striking things that we all agreed was striking from this study was dramatic differences in rates of everyday smoking among our relatives on the Northern Plains versus the Southwest, which we all agreed needed to be followed up, and that this was a good opportunity to delve into this particular kind of a study. But other things we've decided to do when we've given ourselves a nice long opportunity to have a conversation over 6 or 8, 12, 15 months has been to use all qualitative methods to conduct a study with traditional Lakota healers, to conduct an environmental justice study that addressed mercury, increased levels of mercury in surface waters on my reservation. My latest 2 intervention proposals that were sent in just recently was to use a song-based language revitalization intervention among non-socially conversant singers and drummers for the purposes of life promotion. The other study that we've sent in just recently is to test for the first time in a high-income country like the United States, a conditional cash transfer proposal targeting K through 5th grade early childhood school attendance, but intending to measure as outcomes both person level and household level measures of nutrition in the recipient households. So all of these, of the 12 studies that we've conducted so far in about the last 13 years, it's been from 7 different ICs, 7 of the 27 ICs here at the NIH. And it's not principally because it's what I wanted to do. It's what, when we sat down and spent some time together, we all decided we wanted to do. So that's one way to do research. I can do that though, because I work in a community-based setting. It was mentioned that I'm on the faculty of the University of Colorado, Denver, and I am for 2% of my time. But I work for a community-based nonprofit that I founded in 1998 called the Black Hills Center for American Indian Health. And so that's my view on conducting research with our people. So, but if it takes us in this direction, like the study that I told you that we're just wrapping up, I'll go there. But I'll only go there if we're all willing to go there.
So in this panel, we were asked, can health research, genetic research benefit American Indian and Alaska Native communities? And I started out my presentation to say yes, and then I went in to tell you how best to do that. And one of my personal fears is that we lose out. And there's a lot of promise in genetic research, but it has to be done appropriately. And so I went to all of that discussion and in-depth trying to say how we can go about doing that. And my background is I have a PhD in population genetics. At a time where genetics was seen as a promise, that changed overnight. So that genetics became something that was very inappropriate, tribes shied away from it. And so there were lessons there that we can learn. And so I've spent my career really trying to educate tribes about how they can take this tool, but doing it in the right way, making sure that, again, the benefits are really there for the tribes and that it's on their terms, and that tribes recognize that they do have rights, that they have a voice in how this unfolds. It's not a given. It's something that is developing and can be done to the benefit of tribes. And so it's been my personal goal to educate tribes about that process and educating myself about that process. And so it's something that I use every day. And as an epidemiologist, because genetics is too controversial, I now am an epidemiologist, and I've spent my career working with tribes on priorities that they've set, diseases that they want studied, conditions that they want to understand better, and really taking the opportunity to bring Native students on board to help me so that they can have a practical experience of working with tribal communities and doing research the right way. And that's something that I feel is really important, is to take my skill set of what I've learned in school and apply that to help Native peoples and to really share what the benefits of that might be so that other tribes don't have to go through some of those lessons that have really been hurtful to other tribes.
Thank you. We have time for one more question.
Hello, my name is Kate Arroyo. I'm a member of the Cherokee tribe, and I'm a law student here at the University of the District of Columbia. And I just want to quickly address two things. One of the questions from my Cherokee sister about, the researchers and the ethics and the behavior. Some of those things will have to be taken care of and controlled from outside the scientific community, from, legislative, regulations, administrative laws put in place. And that is one of the reasons I'm in law school, is because I know that until our people have a voice, we have no power. And I want to become a voice for my people, and I thank you for having a gathering like this to help educate me, because I'm not an expert on anything, and I'm trying to learn more about issues that face our peoples, and I'm grateful to be here and learn things. One of the first questions that was asked, though, was the issue with not having enough Native American representation in the fields of science, medicine, et cetera, and this specifically genetic research. And the fact is, the solution doesn't come from the problem from the end. The solution starts at the beginning. It is completely about educating our children. When we start getting our children to graduate from high school, to get through college, to get through grad school, that's where our solutions begin. We need funding for programs like STEM, ACES, MESAP, of which I'm a part of myself this summer. I researched for 2 years before coming to law school for funding and scholarships to help me get here, and found very little of Native American funding. There's less than 2% of law students in this country that are Native American. Anything that requires a graduate degree or even a 4-year degree, any of those fields have the same problem. We have a lack of Native American representation because of a lack of education. And I know that sounds like a really simplistic answer because there are so many problems associated with that, but we must focus on that. And corporations and organizations that have money and have programs, they need to extend part of their efforts and time and money to have internships, bring those Native students into them, have scholarships so they can get to undergrad, so they can get to grad school, and it not be such a battle for them. If we want that fruit on the tree, but to do that, we must first nourish our roots.
I have those are good comments and good questions. For those of you that are Cherokee, from the Cherokee Nation in this room, the Cherokee Nation had one of the first tribal IRBs in the nation. It's totally based in the Cherokee Nation, and the name you should write down for connections from the IRB. I know that's scary, people, but we're really here to help you. All the way through the NIH is Sogal Khan. I have worked with Sogal for 20 years. He can get you connected in that Cherokee Nation. It's happening down the road.
So in conclusion of this panel, I wanted to bring up a couple things that have been current themes that we've been talking about. And one of them is can genetic research benefit Native communities? And I think the answer is yes. Everybody has come to that consensus. But with that, how do we regulate, or how do we watch over the experiments, the samples, the tissues, all things that are related to this kind of research? And I think that we've determined that a high level of participation by our Native communities is extremely important. But one of the things that I've heard most of all is when this research comes to our communities, to have a clear, defined statement of what is going to be done and how it's going to be done. And I think that with that level, having certain expectations and having those expectations being met and bound to is extremely important for the research in genetic research in Native American communities. So with that, I want to conclude, and I would give one last round of applause to all of our panelists, and thank you for being here this morning.
Thank you so much. Small gifts from the amazing bookstore here.
And everybody's going to be here for the entire day, so if you have additional questions, please seek them out and follow up with those questions and conversations.
Perfect segue. So we're going to go ahead and break for lunch. If you did have questions that you didn't get a chance to get asked, please engage with our panelists while in between bites of the wonderful bison burger that they have at the Mitsatam Cafe, wonderful wild rice salad. We'll be back. Please be back around 12:20. We're going to start promptly at 12:25 with our next panel. Kayana, thank you, folks.